Today's course is about modeling livestock. So this is going to be a little departure from things we've done before and from things we do later, um, in a sense that I'll explain in a second. I'll start with a sort of remark. Um, I was given this topic to talk about as part of we we spread out the topics. Uh, While I know about the epidemiology of diseases of livestock, I'm not very sure about why I needed to tell you about this. So this was my first remark: is why on earth should I be talking to you about models for livestock? Typically, what I do is I model infectious diseases, their spread, and I've also done some work in what we would call mathematical ecology, in the sense that looking at how species interact. I've even done stuff where we've looked at how filaments assemble, grow in cells. But for me, something like models of livestock is something I've never done. And so, as a word of warning, we're going to be taking a dive into this topic, these models for livestock, um, and as I've said, this is not really my domain. So it was an interesting thing, but it was actually very interesting lecture to prepare. So digging through the literature, there's quite a lot that is done, and some of the models that are made of livestock are actually quite interesting. And so for me, this was a discovery in a sense, and I hope it will be for you too. And before we carry on with the rest of the course, I mean, the disease spread aspect, I think it's actually interesting to take a little detour in this topic. So a few sort of words of warning. The first is that, as I've said here, I've excluded anything to do with infectious diseases. Okay, so there are a lot of models of livestock that actually introduce infectious diseases because it's a very interesting and important problem. But here I decided to take a tour on the pure, let's say, livestock approach. Um, and also, in the interest of time, I should point out that what I did is I was quite lazy compared to what I've done usually, is that I have cut and paste whole pieces of the papers that I'll be talking about rather than redo the figures and write the equations myself. Um, so I hope to give you a little sort of glimpse into this fascinating and quite interesting topic. So what I'll do is I'll cover these topics. I'll talk about first models for cattle because at least as far as my digging through the literature went, this seems to be where most of the literature was. I'm guessing perhaps the way I formulated my Google Scholar query sort of influenced that aspect. Um, but anyway, I think it's a good place to start, and it will provide some topics that I'm sure are easily ported to other animals. So I'll also have a few models for other herds or other species, models for management, and then a couple of words in conclusion. So let's start with these models for cattle. And I'm going to be looking at two different scales. I'm first going to look at a few models that consider um, what happens for individuals. That's going to be quite important. And then we'll switch up to considering models for herds. So at the individual level, I've made this beautiful graph. This is something I found on the web, but I've included a few arrows of my own. The idea is that from a production point of view, because cattle is mostly used to produce either milk or meat, the cattle can be thought of as essentially a system in which you feed something, and there's output that is either in the form of milk or growth of the animal, which is going to result in the production of meat. And there's outflows as well in the form of, and we will see, methane and manure. And all of these are very important topics. I'll just mention in passing, a friend of mine did his PhD at Guelph, which has a very good sort of agricultural component. And his not only his PhD was there, but his wedding took place there. And when you go to, I don't know if it's still there, but when you go to the, I think it's the Grad lounge or whatever, there they have a sort of mechanical cow where you can tweak entries in the form of little dials that you can set to different values, and then it will show you how much growth and how much output is happening because of that. Okay, so this is like from an agricultural point of view, a food production point of view, the cattle is essentially a cow. I mean, it is essentially a device in which you have inputs and outputs. So the first the first model that I want to show is a model that talks about the metabolism of the mammary gland in lactating cows. So this is a model for producing milk, and it's the idea is to try to understand how the metabolism works, and of course, the ultimate idea there is to be able to control milk production in a better way. And you see, by the way, you can see that there's someone from the University of Guelph, as I was mentioning before. They have a very important component there. So this is going to be, if you took the course last term and you saw my lecture about modeling ideas, this is a typical sort of compartmental model type approach. It's not exactly a compartmental model because there's also interactions of other types that are considered, but the type of model that you get is very much a model that looks compartmental. So one of the sort of constants in all the models that we will see is that they are much more complicated than the models we have looked at in the context of infectious disease propagation. There is also a much better understanding of what goes in these models. Okay, so here, for example, on the next slide, I'll show you a sort of what they call a steady state diagram, but I would call an interaction diagram, or it's a sort of flow diagram, although it's not quite flows. But this diagram is going to involve all of those components. So you can see there's amino acids, essential amino acids, non-essential amino acids, etc. There's um, a variety of components that are involved in these models, and also in these in the dynamics of a system like production of milk. And so this is the flow diagram slash interaction diagram that I was talking about. And I'll specify, so it's called here a steady state diagram, although I would not call it this because it's making the assumption that the system is at a steady state, which is not necessarily the case at the beginning. But it's an interaction diagram. So all these quantities that we saw on the previous slide, uh, they interact with each other. So some of them are hormones that regulate or positively or negatively the production of other hormones, etc. There's a variety of factors that are involved here, and this diagram is showing you how these things happen. Okay. The fluxes are indicated here. Okay, so these numbers that you see on the arrows are fluxes, and they're in micromole or millimole per hour. Um, And so you can imagine this is a complicated component. There's a variety of parameters involved as well. And so in the equations that I'll show you in a second, you will see these parameters. So you can see the units are given here, um, and the different notation is given on the left-hand side column. Okay. So these parameters are actually estimated, and their values are given here. Um, and then what you get is a model. This one, I think I am showing you the integrality of the model, and you can see that there's going to be a variety of components in this. So for example, there's plasma flow, and it's influenced by a difference between an input and a bunch of outputs, where the outputs are computed that way. And there's a component here whose dynamics is influenced also by inputs and outputs. As I said, a variety of these models, they always think in terms of inputs and outputs. So it can be inputs and outputs into compartments of the system. Typically it will be, so that's a function of what the cow ingests, okay? But then these make certain chemicals and certain proteins and certain molecules available, and etc. So you can see the model is continuing. There's a variety of equations that are happening here. This is how each of the terms in these equations are computed. There's a compartment here. I I'm, I can't, I think I am not showing you all the equations, but you can see the type of problem that is here. The model, and I should point out, most of the differential equations, if you're thinking of this approaching the problem like a mathematician, most of these differential equations are very simple, they're linear terms. But one of the advantages here is that a lot of the biochemical reactions that are happening are well understood, much more so than when we parameterize a disease propagation model. And so that's why they're able to write down these terms so precisely. And a lot of the constants that I was talking about earlier are also really well known because they represent the rate at which a certain chemical or biochemical reaction is taking place. Okay. Now, that was a model for production of milk. Well, it's not essentially, it's a model for the dynamics of components involved. Now we're going to turn into some of the byproducts of raising cattle. Um, the first one that I'm going to talk to you a little bit about is a model for you can see enteric methane production emission from a non-lactating dairy cow after feeding. Okay, this is a paper from 2015, and okay, methane production is a really important problem. Okay, if you think about climate change, methane makes up about 15-16% of the gases that are emitted by humans. I mean, because of humans. In this case, it's not humans that are emitting them, but these gases are emitted because of humans. And of these 16% of the greenhouse gases that methane makes up, about 40% of that comes from agriculture and to a large extent from cattle. So this is a very important problem, um, especially nowadays. Okay, and understanding the rate at which methane is produced is an important problem. So this is again a very simple linear model, mostly. Okay, so essentially, what they do is that they look at the rate of methane emission. That's going to be that dV over dt, and this is given in grams per hour. And that rate of methane production is proportional to the methogen, methanogen, sorry, that's a word difficult in French, methanogen gas, and the activity also with methanogens and degradable substrate in the rumen. So that's the digestive tract, essentially, of the cow. So it's expressed quite simply like this. But what they do is they actually split this production in two. They separate between residual substrate and newly ingested feed substrate. Okay, so they said they split the volume of methane production into two parts, this V1 and this V2. Okay, so basal and feeding components, and both of these components contribute to the production of methane. And again, like the previous model, this is something that is much better understood uh, than um, whatever you will see with infectious diseases. Okay, so let me show you the variables and parameters that are in this model. So you can see V and V1 and V2 are those quantities that we were talking about. So this is the emission of methane, and as described, as divided between the residual substrate and the newly ingested feed. Okay, and then the derivatives, of course, are the rate of change of these quantities. And then there's a bunch of parameters that are used to parameterize this model. Now, what's very interesting, and we will see this several times during this lecture, is that when you're thinking about diseases of livestock, it is easy, well, not easy, but it is possible to conduct experiments. And so the authors of this paper have done so. And so what I'm going to show you here briefly is how what type of experiments they conducted and how they used those experiments to help in their modeling. So, first of all, the same way, even though it's cattle animals or livestock animals, you need ethics approval. So there was an ethic approval involved. But the idea is that what they took is 16 non-lactating Holstein cows. Um, and they put them in chambers in which they controlled the experimental conditions, both the feed and also the methane production. Okay, so they were housed in a tie stall dairy barn, so that means cows are their movement are restricted, but the cows were used to this context, and they, depending on the individual cows, monitored the gaseous exchange and also the feed to to match the conditions of the cow. Okay, but then they measure everything. So they were um, allocated to a single respiration chamber for 2 days, and then they did measurements. And so here I'm giving a little bit more description. So first of all, um, the diet. These are some description of the diets. So essentially, uh, the concentrate is the part that is important in terms of determining the production. And they had roughage, which was given essentially, you see the allowance of concentrate and roughage decided by the farmer and depends on the live weight of the cow, etc. Okay, so if you go to the paper, you will see the details of this. The concentrate, which in other contexts we would call that a limiting substrate. Okay, so it's the where most of the products that lead to methanization are ingested. Okay, the concentrate was given in at two times. Um, but the roughage, the rice straw was available more readily, and the water was available readily. And in terms of measurement of the emissions, what they do is that they have a chamber that was 3 times 2 times 2 meters. That chamber has a front and a back door. It's fitted with rubber seals so that no gases can escape except where it is collected, so that they can get a very precise reading of the amount of gas produced. So here you have the experimental details. And what they get is a reading like so or like so. So this is the time after morning feeding, for example, and this is the methane emission patterns for the different cows over a 24-hour period. Okay, and it's smaller, so A and B, these are the same, but it's 8 cows, smaller cows and larger cows, okay, from 0 to 200 kilos and from 200 to 400 kilos. The type of readings that they get for the different variables that they use in the model. So this is the body weight. So it's 16 cows, body weights, and a variety of things that are measured. And these are the methane production. And so they use that to parameterize the model that they wrote before. And so what you can see here, the colors correspond to morning feeding and afternoon feeding. Okay, the black is morning feeding and the afternoon is red. And what's most important is this graph here that is showing essentially along the main diagonal that you have there, that would be that the predicted value matches the observed value. in terms of methane production. So the closer you are to that first diagonal, sorry, like this, that first diagonal, um, the better the fit between the model and the observed value. And there's a few values that are out of range, but for the most part, the model does quite well at predicting what the methane output is. Okay. Now let's look at another aspect. As I said, so if you remember my drawing of a cow, there were several components to this. The second important component in terms of the byproducts of cattle husbandry, let's say, is manure. So this here is a model of manure volume and composition production, and this time it's from lactating. Remember, the previous ones were not lactating. These are lactating Holstein cows. And so the model is something like this. So here I'll point out this here is a statistical model. When you see something like this, you know that you have a statistical model. It's, I think, the only one I'm showing in this lecture. I should point out that most of the models, I mean, a lot of the earlier models that you can find were mostly statistical in nature. And then in more recent years, although it's been happening for a while, we also have deterministic or stochastic mechanistic models of the type we just saw before that are also being used more and more. But a lot of literature will involve this type of model. Okay, so you have a variety of variables that are considered, and based on these variables, they write models, and then they use model selection techniques to find which models best fit. So very briefly, this is the type of thing that you would get. So you see, depending on whether DMI is present or not, you'd have a bunch of equations, which here are essentially parameterized already, and this indicates how the different factors are playing. The type of things of components that are looked at. So these are the outputs. Okay, so this is the composition input. Sorry, this is the composition of the diet, and this is the output essentially. Okay, so this is the diet in terms of dry matter, etc. So what is uptaken by each animal in terms of, so this is dry matter, so this is the number of kilos per day. And then this is the production, so the number of kilos of milk produced per day. And then the fecal components, so the manure, how much manure is produced, how much urinary, how much pee is produced by the animals, and etc. And this is measured also for all the animals. And you can see here, this is using a much bigger data set because this is much easier to measure than methane. And so this is used to try to get a model that will predict how these cows will, how much manure and the composition of the manure is going to be. Okay, so now let's switch tracks. And I should point out, we'll come back to some of these issues we just discussed. There's two aspects that I haven't discussed. I mean, one that I haven't discussed, and one that I will mention later. The one thing that I didn't talk about is growth of the animal in terms of weight. I will not discuss it in terms of something linked to the alimentation, I will talk about it. You will see a little later. But that comes in models for other herds. Um, and we'll also look at a sort of tangentially related topic, which is how do you, all that manure that is made, how do you dispose of it? But for the moment, what I want to do is look at herds. So compared to what we had before, we had a single animal. Now we're taking a bunch of animals and we're wondering what happens to them. So I'll start with a very interesting model that is essentially the type of thing that I would be very happy to have written. So this is a modeling paper. This is a pure modeling paper, and I should say it's quite abstract. I don't think, as it is written right now, it would be very usable. It's got a little punish aspect to it, which I'll mention as we go. But it's a really cute model, so I thought it was a good model to describe. As a modeler, this is something that really talks to me. It uses what are called switching systems. I mean, there's a variety of names for these things, but they are systems whose right-hand side, if you're thinking from the point of view of differential equation, x prime is f of x. Well, this is a system where the f of x changes based on the state of the system. Okay. So the idea is to start first with individual cows, and we are going to look at a herd, but we start with a cow. And as I said, this is quite an abstract model. It's sort of abstracted out, but let's see how it works because I think it's really cool. So we start with a single cow, and the cow is represented by three state variables: x, y, and theta. X and y live in the unit segment. So the xy variable lives in the unit square. And theta is discrete. Okay, theta can take three different values: E, R, or S. So E is that the cow is eating. R is that it's resting, and S is that it's standing. So it's essentially, well, it's not essentially, it's assuming that a cow can be in these three states. It's either eating, resting, or standing. And the idea is that their propensity for being in one of those states is driven by these two variables, x and y. So x represents the extent of the desire to eat. So if x is 0, the cow has no desire to eat. If x is 1, the cow is dying with really wants to eat. And y is the extent of the desire to lie down. Okay, so likewise, if y is equal to 0, the cow does not want to lie down, whereas with y equals to 1, it absolutely needs to lie down right now. And as I said, this is a switching system. So what happens is that depending on what state it's currently in, the cow has those two variables, desire to eat and desire to lie down, that changes. So for example, if it's eating, then its desire to eat diminishes as it eats. Okay, so this is the decay rate of hunger. So the cow really wanted to eat, but its desire to eat diminishes with that linear rate. So this model is linear, by the way, okay? It's linear, but it's a switched linear system. That makes it more difficult than a classic linear system. So if a cow is eating, its desire to eat diminishes because it's eating. But while it's doing so, its desire to lie down increases. Now, if it's resting, its desire to eat increases and its desire to lie down decreases, okay, because it's currently resting. And when it's standing, it is both becoming more hungry and more wanting to lie down. So the idea is that when it switches, so depending on how things evolve for the different variables, the cow is going to switch between these states. So let me show you this. So what happens is if I'm resting or standing, and that x gets to 1, and x, remember, is the desire to eat. So if I'm resting or standing and my desire to eat becomes maximal, then I eat. If I'm eating or standing and my desire to eat is less than 1, but my desire to lie down is maximal, then I lie down. Okay. And if I'm either eating or resting and I'm not quite entirely hungry, but either I reach a minimum value of the desire to lie down, uh, or I reach a minimum value of the desire to eat, but my desire to lie down is not maximal, then I stand. Okay. So the way you can represent this is by looking at the different states. And what's going to happen is that when you hit one of the boundaries, so remember that x and y are between 0 and 1. So for each of the different states, so standing, resting, or eating, when I reach one of the boundaries, I'm making the required switch. Okay, so you can see, for example, here I'm resting, but my x reaches the value 1. My desire to eat reaches the value 1. Well, at that point, I eat. Okay, and likewise, if I've eaten enough and I got here, then I will stand and stop eating. Stand is being standing, but not currently eating. Okay. And likewise. And so here they've projected this in a in a two-dimensional configuration, but I think that one is probably easier to read. Okay. But this is a very, very simple model. Okay. So the system is governed by two linear differential equations. The only sort of technicality, and it is not, when I say only, I don't mean this in a negative way. What happens here is that depending on the switch, things evolve. Okay, and that makes the system much more complicated than if it were just two linear equations. And now we had a single cow, but now we take multiple cows. Okay, and we couple them. Okay, and the way it's coupled is by assuming that the cows sort of look around themselves and look at what the other cows are doing. So they're adjusting their behavior to the behavior of their neighbors with some, of course, coefficients that say how elastic that adaptation is. Okay, so the model that they consider actually is this one, and each cow will switch between the different systems, the different states. So, okay, resting, eating, or standing. So this becomes quite a complicated system, and I'm not going to show you the results in detail at all. I'll just show you one particular graph, for example. Here, what they did is they considered two arbitrary, very artificial configuration, one with a ring of cows, where the cows just look at their neighbors, nearest neighbor, and one with a star-shaped pattern of cows, where a central cow essentially uh, accumulates the information, and you will see right away. But as I said, it's that paper is also making interesting puns, because one of the jokes as a modeler is to say, assume a spherical cow. Well, you can see that they're using spherical cows in their graph. And there's another one. Okay, so this is the type of behavior that they observe. So these are the different cows, their states, either eating, resting, or standing, and how these states evolve. So here, when nearest neighbors situation happen, you can see that the situation is um, much more, much less organized. Because I mean, not organized, but because you can imagine what's happening. The sort of information about the state of a neighbor is propagating to the neighbors, and so this is going one and one and one and etc. Whereas when there's a central cow centralizing the information, the behavior tends to synchronize to a large extent, because that once that cow has switched, then all its neighbors tend to do a bit the same thing. And I'll just point out, continuing in the very nice pun-ish content of a paper, they consider grazing bifurcations, which are a type of bifurcations that happen in this type of system, that have nothing to do with the grazing behavior of cows, but the name of the bifurcation is actually quite appropriate in a paper like this. So this is a very cool, very simple in its setup, but very rich, dynamic paper. Quite different from the ones we saw before, probably very difficult to parameterize in real life, but a very fun paper. Another paper, much older paper, oh, well, much older. It's an older paper by two Japanese authors. And here, this is another modeling paper. This one actually has data as well. Uh, so it's a very interesting paper. What they are looking at here is the spatial pattern when cattle are grazing. So it's a bit the same idea as before, but the problem is a little different. So let me explain this. Just the reason I'm showing you this is that what we'll see quite a few times in the paper is what is called the troop length. And so to explain what is troop length, they're essentially looking at a field with a large area, and what they're looking at is the longest distance between any two animals. Okay, uh, so what they're looking only the distance between the far left and far right, but you can think of them as moving along a single line essentially. Animals is, and that's what they are going to call the troop length. It's how dispersed the cattle, the cows are in the field. In their experimental setup, they make essentially a linear field. But in general, they're thinking about something that reduces to this one-dimensional problem. So they suppose that there's a straight line of length theta, and that there are n individuals on this line, and they're located at random points on this line. Okay, and they're looking at the distance from the origin of each of the n animals. So there's a distribution of the location of animals. Okay, so this is a paper that looks at things from a probabilistic point of view. So this is the notation for here. So they're saying that between 0 and theta, because that's the length of the animal, there's a certain distribution which is uniform, and otherwise there's no no cows. So Then they derive the probability density function of of a location of cows, and I'm not going to go into much detail. I'll just go through the main thing. So you see that what they have is so if theta is the length of the line segment and y is the troop length, then they obtain a distribution that looks something like this. Uh, and that's a distribution that they can characterize the expected value and the variance of. Um, and in practice, they look at a deterministic model. So they're assuming that the change is described by something like this. Okay, so this is how the movement changes as a function of how far the animals are. They do some computations. As I said, I'm not going to go into much detail. I see time running by and I still have a lot of things to talk about. Um, and of course, this depends on whether there is attraction or repulsion between the animals, okay? Because the animals might want to graze by themselves or not graze by themselves. Then they deduce a stochastic model for the troop length. Okay, so the troop length varies the following way. So there's that attraction parameter, and they add a little white noise to this. From this, they derive a probability density function that is time dependent. Okay, so because of course this process depends on time. Um, so they derive a probability density function that gives you the probability that the troop length is between the values of y and y plus delta y. So this is that density. And using some standard techniques that are not necessarily easy, but standard techniques nonetheless, they obtain a PDE for the evolution of this density. Okay. Um, they proceed to a bit more analysis, and let me skip over. Again, this is a very interesting paper, but I mean, as a modeler, I really recommend reading. Uh, so this is the type of distributions you get for G. Okay, so depending on the values of the different parameters involved, you get different types of distributions. This is the troop length and the density of animals, like at different troop lengths. Now, what's really interesting, as I pointed out, with cattle or animals, livestock in general, it's easy to do experiments. So here they have experimental data. So what they do is they set up this almost linear configuration, okay, 88 meters times 6 meters. Uh, they set it up somewhere. They have a plant species that is attractive to the animals that they're using. They're looking at 6 Holstein, again, cows. So the body weight is between 200 and 300 kilometers. The width of the pasture is sufficiently wide so that 3 or more cows could walk side to side, so that they don't have to segment one next to the other. Okay, they can actually progress together. And they observed the system for 3 days. And what they get is this type of data here, okay, which shows you, depending on the type of activity that the cows are carrying out, so whether they are grazing, moving, or resting, which in the context of the model I showed before would be resting, standing, and eating. So depending on this, they show what the length of the troop is, and how it changes over durations of minutes. Okay, which means that they can actually identify the parameters of their model. So that would be that the dashed one is a random model that would just use regular assumptions, let's say, and then they can derive a troop length distribution based on the different states. And you can see that moving states, of course, the troop length is larger. Resting state, they tend to congregate more together, and feeding state is somewhere in between. Now, this brings me to the next paper that I wanted to look at, which I'll mention very briefly in passing. This is something from 89, and that's a very interesting component. It's the measurement of the plant-animal interface in grazing research, and that will bring me to a fantastically complicated simulation model that comes later. Okay. So here I mentioned very briefly that there's some very interesting considerations when you're looking at grazing and when you're trying to understand how the animals utilize the space they are given. And so you can see that here they're essentially making the link between the animals and the plants that they're consuming. And if you think about it, the animals live on the landscape, they form a community, they might form a patch. That means they can aggregate. Then they will drill down essentially to the plant level. And then the plant part, they can not eat the entire plant. Okay. Then there's interaction with the soil, of course, because of the plant. And likewise, here you have the other component of this, which is how the different plants aggregate and etc. Okay, and there's a hierarchy of diet selection that happens. And so if you think about it at the landscape level, so that's at the major level, the cows will have to select a place in the landscape. Then there's going to be a plant community that they might hold on to, and more specifically a patch. And then they get to a feeding station, so that's when the cows make their way to a particular place, and they can dig down to a particular plant that they are going to consume. Okay. Uh, another, so that's the sort of preparatory discussion to this, which is a fantastically complicated model. And I'll point out that this is a simulation model only. It's a model of grazing patterns and herbage intake, so to see how much the field in which how much the food will be depleted in a localized place where um, where the cows are grazing. Um, so that's uh, the model essentially. So you can see that the components in it. So there's a variety. This is an extremely complicated model. Um, but it's not studied mathematically at all. Okay, so this is a simulation model, and you can see that there's different components to the model. So there's an external simulation system that is looking at um, a variety of factors. What is the management doing? Management in terms of the farmer. Um, uh, what is the state? Is there a physical barrier? Then there's an internal stimulation system, which has something to do with how, because these are lactating cows, milk cows, so their feeding will depend on what on how much milk they're making. There's a hunger variable. There's a grazing, sorry, variable. I mean, variable component. I shouldn't say variable. And then there's a variety of things that are going into this. So you see chewing time, how long it takes for the cow to actually get the plant itself, the biting rate, the bite volume. This is incredibly complicated. And so, for example, if you're looking at the feeding component, it's even more complicated. So you can see that there's, for example, the type of things that are being intaken, how things are moving between the small and large intestine, how this is being processed biochemically within the animal, etc. If you're looking at even how the cow is feeding, then there is the bite feature and etc. So I should point out that this also when they do models, simulations. Okay, so you have uh, so this is two types of grazing behaviors imposed as a management procedure, and how the not the different components in the cow respond to these different methods of managing grazing. So you can see that they're looking at how many steps are taken per minute for a cow, how many bites they're taken per minute, how much herbage they're ingesting per minute, etc. And that's depending on what they are doing, whether they are so the setup of the grazing, and you can see that they have different periods here that are represented here on the bottom axis. This model has an absolute mass of parameters. Okay, so you can see this is some of the parameters. This is more of the parameters. As you can guess, it is not entirely clear how sensitive the model is to all these parameters. This is not studied in the paper so much. And this, in my view, this could be a little too complicated to draw any inference from this, unless some of those parameters are really well known. But it is an interesting model, but it's very, very, very comprehensive. Uh, and one last thing I want to do about cows is I was talking about manure earlier, and I want to show you a sort of, very briefly, the sort of model that can come from taking into account cattle. And this is a continuous flow reactor for the anaerobic digestion of cattle manure. So this is a chemostat type ecological setup where you put the manure, you put bacteria, and you want to transform it in some way. Okay, so I took one simple example. There's a lot of things about this. This is not livestock modeling per se, but this is connected directly to livestock modeling. So I thought it was worth spending a minute on this. So what they have is they have a bioreactor that has two regions. They have a flow-through region where the manure is sort of fed through. And they have a retention region. So if you think about, for example, water treatment plants, wastewater treatment plants, they will have that's like those big vats that are full of dirty water. That would be what a retention region would be. Flow through would be entry into this, or if there's a variety of configurations. I'm not going to go too much into detail. I'll just point out, in this case, again, contrary to what we have in epidemiology, we have some very well understood processes happening. So these are the different molecules, let's say, biocomponents that are involved, and these are the reactions that are taking place. There's a variety of parameters that are used, and I'll point out that this, if you've ever done any chemostat modeling, this is advanced chemostat. You can see, for example, that they're talking about monosaturation constant, which is something that is very familiar to anybody doing chemostats. And they formulate, so you see there's ionic dissociation equations that are telling you what chemical reactions are taking place. These are concentrations, okay, when you have in a chemical model, when you have the square brackets, it means it's concentration of NA3, for instance. So from these equations, you derive a bunch of differential equations that describe the evolution of the biomass there. Okay, and I'm not going to spend much time detailing this. These are, again, systems that are really well understood, and where some of those processes, compared to what we can do in epidemiology, are really well understood in terms of the timescales or the rates at which things happen. Okay, so that's why these models work quite well. So there's a variety of things that are happening here. You can see that there's a gaseous phase because, of course, when you're degrading, you're processing something like manure, you're making a lot of gas. You can see that methane production, carbon dioxide, etc. And then they can do simulations uh, of the system and observe the output of the system in different conditions. You can see these are here. We're talking about days in the at which the process is happening. Okay, so this is it for at present for cattle. Let me briefly take a look at other herds, other type of animals. As I said, most of what I've just said for cattle holds for all of these. I'm not going to spend a long time detailing them because they sort of amount to the same thing. Uh, One very brief remark is this is some models, and I recommend. So the reason I present some of those papers is that they themselves have reviews of other papers that I think are interesting. Um, so here, this is sow herd management, and so there's some discussion of this type of system. One thing that I wanted to point out here is that something that I didn't discuss, and we'll come back to that when I talk about management models. I should have put this in management models actually. Is that one very important thing is that the life of livestock herd is organized in different periods. So you have to take into account, and you can see here, for instance, um, the type of events and whatever comes as a cycle in sow reproduction. Sow, I think sow. Not entirely sure. The this is quite important in all that type of system because uh, this induces different periods. So you have to assume that when you're doing modeling of this type of objects, you have to assume that there is a periodic component and that it's not only periodic but also decomposed into different periods. So within a single period, like a larger scale period, there are sub parts, and it is important to take into account all of these things. So here you see they have a variety of models that they've looked at. This paper is not very recent, so some of the papers reviewed are not, I mean, the papers reviewed are don't go beyond, I think this is 2007. So essentially things go to about 2004, but you can see the type of models that are used. You see simulation model, simulation of genetic change. People like to find I don't know whether it's appropriate or funny or none of the above, but names that are sort of relevant to whatever they're looking at. So they made a program called Pork Chop to determine optimal cutting strategies. There's a variety of things. Okay, so there's a lot of models, and this, as I said, this is an incredibly rich domain because it is really important. I mean, it is the livelihood and the feeling of many people. This one I found interesting, and this is a very recurrent theme. So I've taken this example, but there were many others. Uh, Coming from a modeling point of view, I find it interesting that the models that I'm going to show you in a minute are used to do this. I talked earlier when I started talking about individual cows. I was saying that one thing that I did not mention was growth. So here, these are models for growth, and interestingly, these are models that you know. So Brody, von Bertalanffy, logistic equation, Gompertz equation, Richards equation. Here, all of these simple models are expressed in terms of their explicit solution. Okay, they're not written in terms of a differential equation or the difference equation they are generated from. Here, all they are showing you is the equation as a function of time. And W here is the weight of an animal. So what these models want to do in this particular case. So for me, for example, the logistic or Gompertz or von Bertalanffy are population level models where we use these models to describe the evolution of the number of individuals in a population. Here, it is not the number of individuals, it's the weight of an individual. Okay. It can be averaged out, but essentially the W is the weight. You could take the mean weight, but it's meant as the weight. And so what they do is they look at so weight inf is the beginning weight, and the W infinity is the weight when the animal has fully grown. And they're looking at, for example, the different inflection points, because this is something that can be measured. If you follow the weight of an animal, you will see at what point things vary. And so you can see that they used a variety of animals. So these are data that they got in a variety of papers, and you can see there's bulls, cows, pigs, sheep, goats, rabbits, chicken, quail, etc. And what they did is they fitted those different models to these different data sets. And what you can see is something like this. Okay, so the Japanese quail, for example, how the weight in grams vary as a function of the age in days. Some chicken, some guinea fowl, and what they did. So you have rabbit here. bull, etc. This is goodness of fit, so the R squared, the residual mean square error, the Bayesian information criterion, the Akaike information criterion, and so what they're showing you every time in bold characters is which of the models. So if you remember, equation one was Brody, etc. Which of these five models gives the best results in terms of fit for the different population? Okay, and there's a variety. If you go to that paper, you will see that they've done that for a variety of different animal species, etc. And so they show you how to do how this. So you see California rabbit, goat, weight in kilo, sheep weight in kilo, pig, etc. So it's not only quails, etc. And the fit depends a little bit. So you can see, for example, that the sheep is not working quite as well as the others. And let me show you in particular, for example, the same type of approach used for chicken. So this is in Nigeria. The previous paper, they used a lot of data sets from a variety of papers. This here, these authors looked at specific, so that type of chicken, um, several types of chicken, I think, but they also generated data. That's why I wanted to point that one out. So here they used a little variation on the thing we saw before. They used Gompets and Logistic, but they use two different versions. One version that uses 3 parameters and one version that uses 4 parameters. Okay. What they do is they have both. So in another context, we would call in vivo and in vitro. In vitro is when you control everything; it's a controlled experiment. In vivo is when you observe in life. I mean, so in a human, for example. And here it's a bit the same. So they have an experimental setup where they have almost 2,000 chicken um, that they looked at in a very controlled environment. And then they also had on farm tests, and they looked at much more chicken. So we had almost 60,000 chicken that were distributed to 2,100 households across 5 states in Nigeria uh, to sort of get a sense of how things happen outside of the lab. So they have both the sort of very controlled experimental setup, and they have a sort of real life setup. And I'm going to skip on this very briefly, but refer to the paper if you're interested. They do the same type of analysis. They look at different types of chicken and they compute the different parameters, the information criteria, so Akaike and Bayesian, the residual mean squared and the adjusted R squared for these different species. And so you have, for example, female birds raised on station from 0 to 20 weeks. You have a variety of cases that are reviewed in the paper. Okay, the other thing I wanted to talk about is heterogeneous hoods. So I've now set up a heterogeneous hood, and I'm not going to spend a lot of time talking about this, but I wanted to present a model where things go larger. Um, so this is a paper of Mongolian livestock populations. Um, Point out that it involves Nakul Shitnis, who is someone, if you've done any malaria, for example, Nakul has done some very interesting work in that domain as well. So I was actually surprised pleasantly to see that he's also doing things in this domain. So here, this is a discrete time model. And it's looking at the total population. Well, I mean, it's looking at the population, but in particular, it looks at population of goats, sheep, cattle, and horses. And for example, cattle and I believe horses also have juvenile populations and adult populations. So it's further divided into juvenile and adults. Uh, if you look at, for example, a given given population, so if we look at goats, there is a dynamic for the goat population. You can see that in each case, there's so these are the age classes. I'm not entirely sure if I remember what is Y and X, but these are age classes. You have, um, oh, it must be female and male. And there's slaughtering, losses, there's migration into, this is how they age, and this is births. So there's a model like this for each population, and you can see, for example, for goats, uh, we have dynamics for the different goat populations, have a different compartments, so the age groups of male and female, I'm guessing. Uh, Yes, exactly. The expected number of female offspring. So there's a variety of parameters that are there, and then the model is used with data as well. That's observed data. So and so they fit the data and try to use it. Finally, I'm going to briefly go over models for management because so far what we have seen, well, I mean, there was a little hint of this when we were talking about grazing, especially that second very detailed grazing model. But altogether, there are models that try to look at the entire management aspect of a livestock system. Uh, First thing I want to point out, this is a review. I'll just briefly mention it here. This is a review that talks about systems approaches to beef cattle production systems using modeling and simulation. So if you go to see this paper, this is all I'll say about it. It's quite interesting. It'll give you some sense of what was being done back in 2010. Another paper from 2010 that's a bit the same idea, but that's also very interesting, is application of math modeling in beef herd management, and there's also review. Um, and here, what's interesting is I took this figure from there. So they'll point out they have livestock models, so you can either try to optimize or simulate. If you're doing optimization, you might be stochastic, probabilistic optimization, or do some linear programming. Or if you're doing simulations, you might be doing deterministic simulations or stochastic of different types. Okay, so um, I refer to that paper for details. And let me just finish, I believe, with this paper that's the result of a symposium, and that's much more recent. Okay, it's from last year, and that talks about mathematical modeling in animal nutrition. Um, and there's this notion that well, it's got different names, but one of them is precision livestock production, which has to do with the use of a variety of modern technologies, whether it's surveillance, monitoring, or modeling to op- optimize essentially animal nutrition. Um, So the overall idea is this: the what they call a conventional producer decision process involves these. You have real-time data that goes into modeling. This influences the management. You're looking at system performance. This also influences the management. The producer experience influences management, etc. So there's this loop essentially that is used to conceptualize how precision production is organized. And so they have precision systems modeling. I'm always interested as a modeler uh, when I see large scale paper, I mean papers that are address in important issues, and that you see the word modeling coming into here, because modeling is just one of the components of this precision system management. Okay, so you can see, so the conceptual diagram of the different aspects involved in this. And so, for instance, how um, this would organize. You can have continuous grazing, essentially. So this is showing you how information, so decision flow, information feedback, implementation of precision management, where that comes in, and how things are organized this way. So here in this case, you have seasonal information. You take some decisions, and based on these decisions, you attribute the cows, which in that case, continuous grazing is you just put them on the field and see what happens essentially. Rotational grazing introduces locations. So you will put the animals on a place where they can feed, and you've excluded them from other places, while these other places are regenerating the resources there. And then you could have precision grazing where you're actually directing the animals in a much more precise way towards where you want them to feed. And this whole process is uh, quite complicated. Okay, so uh, precision technology means, okay, you're able to monitor exactly, you know where the cow is, and you are able to essentially drive it to somewhere else. Drive like this, but move it to some other place. And you can see that this whole organization is quite complicated. Again, these are sort of conceptual models more so than actual models, but they're important. And here you can see, for instance, um, the type of model that are used. So depending on what type of target you're looking, and then you have, so for example, us as modelers, we'd look at data-based mechanistic, for instance, or semi-mechanistic, but there's also some gray box, which are more learning, more recent approaches, and how these things, the type of response that is observed. Okay, and let me finish here with a very brief conclusion. So I hope I've convinced you that these are very interesting models. And as a mathematician, as a modeler, but also a mathematician, I'm actually tickled by the idea that a lot of these models have been formulated and used mostly from a simulation point of view. And I think that there are potential opportunities for work to be done. So I showed you two very nice mathematical models, but there's another sort of more ad hoc models, and all of those have not been analyzed. And so there are opportunities for people to do things here. I did show only one statistical model, although they make up most of the models that we actually see in the literature. One thing that is very interesting here is that contrary to disease modeling, whether even it's in livestock or not, here producing data in a controlled manner is, I'm not going to say easy, it's a lot of work, but it is doable. Okay, and it's probably to some extent data that you can easily reproduce. Experiments can be run over and over again, and they should give you essentially the same result. So this is like a model ecosystem. One of the reasons I talked about the chemostat briefly when I was talking about the reactor model, one of the reasons chemostat models are so popular is that they are really good artificial ecosystems. And here it's a bit the same. You have some good control over the type of system. And the data, you can make really good data. And finally, I did, as I said originally, exclude disease models. This, of course, is a very important component in there, but because I'll be talking about disease models in the rest of this course, I thought, okay, I'll leave that for later.